Receptors on the natural killer cell surface that regulate the recognition phase consist of two types. One is composed of the receptors that are homologous to the C-type lectins and is called the natural killer receptor complex. The other is composed of immunoglobulin-like domains called killer cell immunoglobulin-like receptors, also known as KIRs. These are shown on the natural killer cell surface as the forms KIR3D and KIR2D. However, the predominant control of the natural killer cell activation occurs through the C-type lectin receptor, CD94-NKG2 heterodimer. One complication in understanding the natural killer cell activity is that the same structural families, that is the CD94-NKG2 natural killer receptors, contain both activating and inhibitory receptors. The NKG2 proteins differ for the inhibitory NKG2A and NKG2B, and the activation NKG2C and NKG2D protein structures of the CD94-NKG2 complex. Signaling by the inhibitory natural killer cell receptors suppresses the killing activity of the natural killer cells. The receptor for the target cell is the major histocompatibility complex 1, also known as MHC1. Natural killer cells will not kill healthy, genetically identical cells with normal expression of MHC1 as seen in other normal cells of the body. Therefore, the binding of the inhibitory natural killer cell receptor to normal MHC1 sends a negative signal to inhibit signals from the activation natural killer cell receptors, which are bound to the target cell activating ligand. However, if the MHC1 is 1, not dense enough on the target cell, or 2, if the target cell has processed antigen and displayed it in the pocket of the MHC1 illustrated here by the shaded red-yellow ball, or 3, if the conformation of the MHC1 proteins have been sufficiently altered, noted here by the stripped MHC1 proteins, then the binding of the altered MHC1 to the inhibitory natural killer cell receptor will send a positive to the activation natural killer cell receptor complexed to the target cell activating ligand. It is this positive signal that initiates the remarkable cytotoxic machinery of the natural killer cells.